Okay, this is the second prep lecture recording for lecture 24. And I finished a little bit early because they are doing renovations in and around where my office is hiding, and there was too much noise, so I turned it off for a few minutes. But we're back, and I should have just tried to get through the final slide because this would have been the end anyway. I just wanted to review TGF beta signaling. We have talked about it before, but it has some extra included information. So um, uh, there's a protein, nodal encodes a protein, and that protein has to get cleaved by a pro-protein convertase, so that just part of it that's going to form the ligand is available, and the ligand dimerizes. Okay, and I've talked about the fact that we have heterodimer ligand receptors for the ligand, and that upon binding of the ligand, you have this transphosphorylation of the receptors, and then when they're both activated, SMAD, in this case 2 and 3, one or the other, are phosphorylated, and they bind to the cofactor called SMAD4, enter the nucleus, and they activate target genes. So notice that some of the targets are genes that negatively regulate or interfere with nodal signaling. But nodal is also a target gene. And so what this means is that a cell that has received the nodal signal will start expressing nodal. It will also start expressing the repressors, or the antagonists of nodal signaling. And if you go back to this slide where I was talking about the primitive streak begins with nodal being expressed in these cells. But nodal is a signal, so the signal is going to move to these cells and activate nodal expression. But they're also going to activate targets that shut off nodal signaling. So you're going to see, and as gastrulation proceeds, the cells here that stop expressing nodal, they're going to start expressing other genes, and you're going to see the mesoderm and endoderm cells moving in, and so on. And there'll be this wave of gene expression changes, which is like a wave of nodal sweeping along. So it activates the cells nearby to express itself and the inhibitors of its signaling. Right? So that's how you get this wave of nodal expression starting at the posterior, activated also by FGF, and then that wave kind of transfers forward. Okay, and I'm going to leave this part out. This is another level of regulation, but we're probably not going to touch on it this time. And so what do Cerberus and Lefty actually do? They are proteins that Lefty inhibits the activity, the binding of nodal to the receptor, as well as the actually activation of the receptor. And Cerberus basically just interferes with the production of the nodal ligand. So these are your negative regulators. Okay, and that was really all that I was going to talk about for this particular part of the chick embryo. So the mouse embryo is also quite regulative. It's probably not as regulative as the chick, but you don't see any potential axes in the mouse embryo. The earliest embryo axis is the embryo and ab embryo, which we'll talk about in a minute. And also, you know, within the blastocyst here, um, you have, you know, the inner cell mass, which is going to form, some of which is going to form the embryo, and the ones on the outside are going to form the placenta and other extra embryonic structures. So there's a whole lot of additional extra embryonic structures that are starting to be specified early. But you can actually move cells. So this is looks like an 8-cell stage. You can actually take a cell that's from one embryo and embed it into another embryo. And if this cell most of the time forms trophectoderm in its original position, when you move it to the new position, sometimes it still forms the structure it was intended to form, because there's a lot of regulation going on and the cells can shift around a little bit in here. But you also see a change in most of the cells to form an inner cell mass cell. And so this means that cell position determines, to a certain extent at least, in the early stages, whether um, the cell is going to form trophectoderm or inner cell mass, right? And then by the time the blastocyst stage has happened, you have CDX2 transcription factor that is expressed in the trophectoderm specifically. And then you have OCT4 in the inner cell mass. But then OCT4 activates expression of two different genes, which we'll talk about in a minute, that distinguish the primitive endoderm from the epiblast, which is the embryo proper. And before we get to the 32 cell stage, which is you know, a couple of cell divisions past this 8-cell embryo, the fates are not determined. So as late as this 32 cell stage, you can still move the cells around and they will change their fate. Okay, so this is an important point, and that is one of the reasons why we say this is still, it's not as regulative as the chick embryo. You can't divide the embryo into pieces at the 20,000 cell stage and get lots of little teeny weeny embryos all developing properly. But it is still quite regulative. So we have no determination until the 32 cell stage. They develop depending on position. You can do these kinds of manipulations. So, you know, you take an outer cell, you implant it into the inner cell mass of a different embryo, and for a lot of the experiments, you will see that it will participate in formation of the inner cell mass. You can take a whole embryo and surround it with blastomeres from a different embryo, so that you have, you know, one embryo kind of inside another embryo, and you will get some reorganization, and a large embryo forms, and it will make one specific embryo that's properly patterned, and so on. And you can also take, so if you had a couple of embryos and you collected up all the inside inner cell mass cells from the two of them and put those together. Remember, I've said this a few times, but embryonic cells have a lot of sort of cadherins and other cell adhesion molecules on their surfaces. They are what we call very sticky cells. So you can take, like for mouse embryos, you could take embryos of two different genotypes of mice with two different coat colors, and then you can sort of aggregate them together in vitro and then implant them into a female, and you will get a mouse that has developed into a perfectly normal embryo, and mouse, a baby mouse, but it's composed of two different genotypes. So some of the cells are from one original embryo and some of the other cells are from another, and you can see this visually by seeing the different coat colors and that sort of thing. So because of that stickiness, you can do this kind of experiment where you take all the inner cell mass cells from two different embryos, put them together and culture them, and they will reorganize and develop into a normal blastocyst. So some of those cells will become the trophectoderm and some will remain inner cell mass. And you can also take the outside cells, the trophectoderm type cells, and stick those together, and they can also rearrange, reorganize, and develop into normal blastocysts. So this is what we mean when we talk about regulative development. The fact that that negotiation can occur. Okay, so we're just going to look at a couple of genes here. So there's 4 genes to know. And when we look at the 8-cell stage, the 8-cell stage embryo, these 2 transcription factors are expressed at very low levels in all the cells. And you will remember that I mentioned back here that these are actually expressed in distinct subpopulations. So we'll talk about how that works. Now, so low levels, but when we get to the 16 and 32 cell stage where we've had the cell division, and the cells with the tight junctions are on the outside, and the cells with the adherens junctions are kind of on the inside, we see a differentiation. And so CDX2 is inhibited by OCT4 expression. So the inner cell mass starts expressing OCT4 in higher levels, and 2 of its target genes, NANOG and GATA6, at low levels. Okay, and so CDX2 is limited to just the outer ring, and so that's specifying trophectoderm. And then later on, some of the cells in the inner cell mass start expressing NANOG, and some express GATA6, and it's almost like a stochastic process in a way. Whichever cells are expressing more of one or the other, they start expressing exclusively that. So all the blue cells were expressing little amounts of each of these 2, but they ended up, as NANOG was expressing at higher levels, then GATA6 was shut off, and vice versa for these cells. And when they sort out so that all the NANOG expressing cells stick together to form the epiblast, and then the ones that are expressing GATA6 all sort together. They stick with each other, and they form the primitive endoderm. So we get this sorting out of cells, and it's based on which of these genes is expressed at higher levels, but these are having effects on adhesion properties. And so these cells will stick together because they're expressing now similar cell adhesion molecules on their surfaces that bind each other preferentially, and these guys have cell adhesion molecules that are holding them together preferentially as well. Okay, and these mechanisms are largely conserved. One of the things I didn't include in our human genetics lecture was a slide that looks a lot like this. But is also is talking about human embryo development, and these are the same exact genes that are expressed in these early stages in the human, and largely conserved in pretty much all the mammals that have been, rabbits and guinea pigs and so on. Okay, so that's the first part. That's how the trophectoderm is specified, epiblast and primitive endoderm. And so I want to talk a little bit now about, so this is a 6.5 day embryo, and at this point the primitive streak has started to form with this thickening of cells, which, remember, is because cells are migrating away from this side and moving in towards here, and then the cells are also, as the thickened region sort of moves down here, or it shows up down here. Remember that the mesoderm cells are moving in between the ectoderm and the endoderm, which are kind of reversed. And that the beginning of the primitive streak, so the first sign that you can see it is 5.5 days, and we can see it to mark the posterior pole. And this happens a little bit later. I have day 15 here, but I'm pretty sure I read it was day 14. I may have typed that in incorrectly, but it's a little bit later. So after the blastocyst formation, which is only, and the beginning of implantation, which is only a day or 2 apart, in mouse and human. After that period, human development takes considerably longer proportionately than mouse development does. Okay, so we want to go back and look at slightly earlier stages and just see, there's some movements of cells that kind of initiates, precedes the appearance of the primitive streak, and this will help us understand kind of anterior, posterior. So here's our epiblast, which is our layer of cells. The embryo is cup-shaped, okay? And this is 5.5 days. So this is just barely when we might start seeing a very slight thickening at the posterior pole. But yeah, this kind of precedes that just a little bit, okay? And so this is the distalmost. So this is the tip of the cup, and the cells there. Remember that the endoderm is on the outside of the embryo at this point, and the ectoderm, mesoderm is kind of on the inner region. And so this is called distal visceral endoderm. Okay. And the epiblast sends nodal signals, and it sends it in all directions. But when this batch of cells receives it, they start expressing anterior specific genes as well as proteins that are like Cerberus and lefty. Remember, we just talked about this. These are inhibitors, basically, of the nodal signaling. Okay. So then these cells start to divide. That's what proliferate means, that they divide more. And they start to move anteriorly. So they move up in this direction, and then they become anterior ventral endoderm. Okay. At the same time, this is the extra embryonic endoderm up top here. This is already starting the process of implantation, and this is extra embryonic. It's not going to contribute to the embryo, but is going to participate in the formation of membranes and placenta and all that sort of thing. They're sending BMP signals, and the BMP signals are basically preventing the anterior visceral endoderm from migrating any further. It can't go way up here. It's held to a limit because there are these BMP signals coming from this region. So the signals to know are the nodal signal, Cerberus and lefty, which I'm going to show in the next slide, and then the BMPs. So this, again, this is at 5.5 days. And then by 6 days, the anterior ventral endoderm has migrated all the way around. And it is starting to send these signals, the lefty and the Cerberus, and they are inhibiting nodal signaling. And that is causing the ectoderm here to form anterior ectoderm. And that's going to specify eventually the forebrain and the anterior neural tube. So before the primitive streak actually shows up, which isn't until 6.5 days, there has been a bunch of signaling and so on. And so this region already, quote unquote, knows, okay, we are anterior, and this is going to be the forebrain, right? And then the migration of cells that move, not the ones that are going to form the brain, but other ones to start forming the primitive streak, that takes place at 6.5 days. Okay. And the visceral endoderm, both distal and anterior, they are repressing the formation of the primitive streak in the anterior, right? And so it's forming in the posterior, and this is another example of potential default state. Okay. And I think I would like to stop there again because these are relatively simplified versions of things, but there's still quite a bit of information, and probably good to have another separate video for the rest.